Tessie. Okay.
The green light is going.
My voice.
Correct. I didn't turn on my focus.
Okay, cool. I like your central location. Huh? 1 minute? Okay. Wow. You're advanced now. You want the mic, Karen?
I don't think so.
Okay.
What's that? Craig Robinson.
Oh, look at that. Oh, yeah, she's in town, man. Hello.
So, but you are going to wear it when you present.
Yeah, and it's on right now, so people on Zoom can hear us. If you
Okay.
want it, you can
Or you could
have
just,
it for a minute. Or I
I
can
mean,
stand
you
by
could stand over here. It would be awkward if you could zoom in this.
Okay.
Because, you know. All right, let's get started here. I'm delighted to welcome you to Montana's. Sorry, I must have we got back-to-back defenses here. To Montana's defense. Montana is a banana slug. I don't know if you are aware of that. She's a native Californian. This is great. She's going to act out what I have to say. She's got a BA in Earth Science and Physical Anthropology from UC Santa Cruz. So when she applied to this position, she had extensive experience in wetlands and springs, and that was particularly exciting for me, including a position that she had most recently with the Spring Stewardship Institute in Flagstaff, and she was conducting ecological surveys all throughout the American Southwest. And before that, she was a restoration specialist and, I gotta get this right, a subterranean technician for Bats Conservation International, also in Flagstaff, where she planned and executed all sorts of wetland and riparian projects. It was actually really her exceptional horticultural and plant propagation background, though, that made me very excited about her skill set, including some professional experience as a seed farmer and horticulturalist, which, you know, I don't get many applications like that every day. So when she applied for the lab, I really knew she was the right person for this potential project you're going to hear about today, given her passion for botany and seeds and plant propagation and restoration. Montana has become a very highly valued member of our lab. She gives clear guidance for her humble demeanor, and she's done a great job mentoring quite a few undergraduates as well, sharing her passion for botany and seeds. She also shares my family's love of produce, so she's been a regular member of the harvesting group in my yard, in my garden, in my house. So a few months after Montana arrived, she commented to me that I had done her a solid by pairing her with this project. It took me a few minutes to actually understand what that meant. That's a sign of my age. And then, I realized we had this moment of, right, I understood where she was coming from, her experience, her interest, and her passion, and I knew what was needed for this project. And I think her passion for this project really comes through, as you're going to hear about today, about drought-tolerant plants of Great Salt Lake wetlands and some of our interesting experiments, really helping address some of the biggest issues we're facing with wetlands in semi-arid regions. So I will turn it over to Montana.
Thank you, Karin. My name is Montana Horchler, and I'm in the Wetland Ecology and Restoration Lab. Before I start, I just want to thank everybody for being here, and I hope that you find something interesting in my talk today. And if you're a restoration practitioner, I hope you find something that's potentially relevant to your work. I'm just going to check my mic is on here, and it is. Okay, so I'll be presenting today on drought-tolerant plants of Great Salt Lake, determining dormancy break, germination, and seeding timing requirements to inform restoration under hydrologically extreme conditions. So this is my outline here. I will first talk about the research context, then get into my growth chamber experiment and my field experiment, and then the implications of my research. So the research context. Wetlands are of outsized ecological importance globally, but large-scale wetland loss and degradation are ongoing worldwide. This graph is from the 2025 Ramsar Global Wetland Outlook Report, and it shows wetland loss since 1700. And then this chart here, in North America, introduced and invasive species are a major threat to wetlands. So, Great Salt Lake, zooming in to a closer look at the context of my research. Great Salt Lake is drying and its water future is uncertain, largely due to agricultural diversion, but coupled with increasing frequency of droughts and warmer temperatures due to climate change. It's also an incredibly complex and highly managed system. As you can see on the right there, those are what the various impoundments, the wetland impoundments at the lake look like from satellite view. And those impoundments are allocated water throughout the growing season and throughout the year in different amounts and managed by wetland managers. And they contain various wetland habitats within them. So on the right, emergent and submergent wetlands. So wetlands where vegetation is growing up and out of the water. Playa wetlands, which are flooded part of the year, but dry for much of the year, and meadow wetlands, which are also flooded for part of the year as well. So Great Salt Lake is also facing serious degradation due to an invasive grass, Phragmites australis, which covers large areas of the highly managed system. I'm not on that slide, sorry. Here we are. So Phragmites australis, you can see that it covers large areas of this managed system. And there's an ongoing removal effort by wetland managers, which has left large swaths of the wetlands devoid of vegetation, and they need to be revegetated with native plants to reestablish ecosystem function. Native plants are not going to just come back in this context because Phragmites has been invaded for so long that the natural wetland seed bank is depleted. Yeah, so seed-based restorations have emerged as an appropriate response to the scale of revegetation needed at the lake, and those are ongoing by wetland managers. Historically, attention has focused on restoring wetter emergent habitats that need water year-round, and that's what most of the seed-based restoration efforts so far have been focused on. But with the increases in frequency of drought due to climate change, as well as continued water diversion for human use, managers are seeking more information on playa and meadow species of plants. And in order to incorporate those species into restoration efforts, they need basic information on dormancy and germination requirements of plants from these habitats, as well as information about how to incorporate them into ongoing seeding efforts. So in order to help address these restoration challenges at Great Salt Lake and similar places in the broader Great Basin and Intermountain West USA, I performed 2 experiments. In a growth chamber experiment, I explored the effects of seed dormancy break, germination temperature, and light requirements on the proportion, rate, and timing of 10 drought-tolerant wetland plants native to the Great Salt Lake wetlands, and those are pictured below there. And then in a field experiment, I assessed the effects of seeding timing on native and invasive species cover over the course of 2 growing seasons. With the information from these 2 experiments, I hoped to inform native plant growers in their first step of growing plants, which is seed dormancy break and seed germination. Need to know how to do that to grow the plants. I hoped to facilitate the incorporation of greater species diversity into existing restoration efforts. I hoped to improve the understanding of how seeding timing affects plant community establishment in a restoration context with a focus on these playa and meadow species. And I hoped to inform species selection for wetland restorations occurring outside of emergent environments. So moving on to talking about my growth chamber experiment. And this experiment asks the question, what are the dormancy break requirements, light requirements, and optimal temperatures for germination of these 10 drought-tolerant playa and meadow wetland plant species? Seed dormancy is a state where viable seeds don't germinate even under favorable environmental conditions. This is often due to physiological factors that inhibit germination, and the most common type of seed dormancy is physiological dormancy. And in this type, dormancy depth may be reduced or dormancy may be broken in several ways, 2 of which are warm after ripening and cold moist stratification. So in nature, cold moist stratification usually happens under conditions like winter. So the seeds will sit out and experience cool temperatures and moisture for a period of time and then germinate at some point in the future. That affects when seedlings emerge, such as at the start of a growing season, after winter cold stratification, or before the onset of winter if they don't need cold stratification. Similarly, germination timing and setting is influenced by temperature and light. These drive seed germination seasonality and location. For example, are seeds germinating in canopy gaps? Are they germinating under mulch? Are they germinating when they're buried? Are they germinating at the beginning of a season when it's cool, or in midsummer when it's warm? Germination timing also influences seedling survival, community assembly, and competitiveness with invasive species. And wetland species vary widely across taxa in their dormancy break and light and temperature responses. Though there are patterns in dormancy at higher taxonomic levels, species variability is still high. Species level variability is still high. And understanding individual species requirements for fast, high proportion germination is very crucial for making appropriate choices about how to seed in the field and also how to propagate native plants from seed. Oh my gosh, I'm not moving forward. Sorry, you guys. Here we go. So my experimental design, I had 7 dormancy break treatments and 10 drought-tolerant plants pictured here. I'll go through these dormancy break treatments just to introduce them. I had a no dormancy break treatment in light, so seeds were not subjected to any dormancy break, and any pre-germination treatment before being put into growth chambers. I had no dormancy break, but seeds were kept in darkness and prepared for growth chambers in darkness using a green light in the dark room. A warm and dry after ripening treatment in light, where seeds were put into jars and then stored at 40 degrees Celsius in a growth chamber for 30 days. 4 weeks cold stratification in light treatment, a 4 weeks cold stratification in darkness treatment, an 8 weeks cold stratification in light treatment, and an 8 weeks cold stratification in darkness treatment. And this was crossed with cool germination temperature, which was chosen to reflect current spring conditions in the Great Salt Lake area. A moderate germination temperature, which was chosen to reflect current midsummer conditions in the Great Salt Lake area, and a hot germination temperature regime, chosen to reflect midsummer temperature conditions projected in 2070. These oscillated between a higher temperature and a lower temperature, and during the higher temperature time, seeds were also exposed to light, so that's mimicking day and night conditions. So kind of zooming in more on what this looked like in practice, we had wild collected seed that was stored at room temperature. And then in our treatments, we had 3 treatment types, one of which was the seed dormancy break treatment. We had untreated seeds, cold stratified seeds, warm dry stored seeds. And to the right there, you can see that's the big refrigerator that we stored the cold, moist stratified seeds in, and then jars to represent how we conducted our warm, dry, after ripening treatment. Then we had seed germination treatments. So untreated and cold stratified seeds were then those, there were counterparts in light and in darkness of both of those. The warm dry storage was only in light. And then we had a seed germination temperature, cool, moderate, and hot. And on the right there, that's what a growth chamber looks like. It's kind of like a large refrigerator, but you can program humidity, temperature, and light, and program it to oscillate and cycle. Data collection. Over the course of 28 days, we counted and removed germinated seeds from light-exposed trays, so seeds that had a radicle emerging were removed every time we did our counts. At the end of the experiment, we counted all germinated seeds that were in the dark treatments. So the dark treated seeds had been wrapped in foil to protect them from light exposure in the growth chambers. And then we replicated this whole process 3 times. We predicted that most species would require a period of cold stratification to germinate to a high proportion quickly, that some species would germinate faster, and to higher proportions under warmer temperature regimes in light, and that responses to dormancy break and germination conditions would vary by species. This is just a little taste of the effort that went into this experiment. So it sounds kind of simple when you just talk about it in terms of 7 dormancy break treatments and 3 temperatures and 10 species of seed, but just taking one species here, Salicornia rubra, and then looking at one growth chamber. Each of these cells down here represents the 7 dormancy break treatments, and that equates to 100 seeds that were prepped to go into germination. So for each growth chamber and for each growth chamber, for each species, we had that many hundreds of seeds. And then for each species, we had that many hundreds of seeds in our growth chambers. And then over the course of the experiment, it was that many hundreds of seeds. It was 63,000 seeds counted over the course of 3 experimental rounds. It was a great effort by the Wetland Ecology and Restoration Lab. Onto my statistical analysis. So in this study, survival analysis provided a framework to quantify both the timing of germination using Kaplan-Meier curves and the relative rate of germination among treatments using Cox proportional hazard ratios, which properly account for seeds that never germinate. It's a time to event analysis. Species, we used a global mixed effects model to see how different treatments, alone and in combination, affected the final proportion of seeds that germinated. Cumulative germination proportion was analyzed across species, a combined dormancy break and light germination treatment, and temperatures, with block and growth chamber included as random effects. The final proportion of seeds germinated was analyzed using the beta distribution with a logit link. And these are some of the results. So I'll just run through some examples with you of individual species, because I'm afraid I don't have time to go through every single one. But again, we were looking at dormancy break, light, and temperature, and their effects on germination proportion. In these plots, you can see that each of the panels represents a different species pictured on the right of the plot. The y-axis indicates a proportion, modeled proportion of seeds germinated from 0 to 1. And then the dormancy break treatments are on the x-axis. So the far left is our control light treatment. The next one is the counterpart control dark treatment. Then we have the warm and dry. Then the 4 weeks cold strat and 4 weeks cold strat in the dark. And then 8 weeks cold stratification in the light and 8 weeks cold stratification in the dark. Each of the colors represents a different germination temperature. So blue is the cool germination temperature, green is the moderate, and orange is the hot germination temperature. Looking at the effects of the dormancy break treatments on individual species' final germination proportion, we can see that Disticula spicata, dormancy break was necessary for high proportion germination, including Disticula spicata and Glaucus meridema. So on the left, you can see that before dormancy break treatment for the control dark, and the warm dry, there was low final germination. And on the right there, you can see that at least under some temperature regimes, germination was increased to a high proportion. However, for one species at least, Allenrolfia occidentalis, dormancy break did not have an effect on final germination proportion at any temperature. Looking at light, for Salicornia rubra, you can see that this species was definitely affected by light, and treatment combinations in the light germinated to a much higher proportion than dark treatments did. Same was true for Sicivium verrucosum. Both of those are playa species, interestingly. And it was also true that some species some species did become easier to germinate in the dark after a period of cold stratification, as can be seen for Salicornia utahensis here. So on the left, you can see that the control and the control dark, there's a large difference in final germination proportion between those two. But then after 4 weeks of cold stratification and 8 weeks of cold stratification, the seeds germinated to high proportions under all temperature regimes. Looking at the effects of temperature, some species germinated to higher proportions under certain temperature regimes. So Glaucus meridema, let's see if this works. Oh, it doesn't work on the screen. Glaucus meridema germinated to higher proportions under the cool germination temperature. And the same was true for Puccinellia nataliana. So the hot temperature regime did not improve germination of Puccinellia nataliana, and it germinated to higher proportions under the cool and moderate temperature regimes. So survival analysis is an approach used in medical studies, usually, hence the term survival, but it's also useful in modeling seed germination because it's a kind of time to event analysis, and that's what germination studies try to get at. How long before a seed germinates? What's the probability of a seed germinating at a specific time? And I had two parts to this analysis. The first part was generating Kaplan-Meier curves. So these are Kaplan-Meier curves. They're similar to what you would see if you were to graph the raw germination data over time. So on the y-axis, we have our modeled proportion of germinated seeds. On the x-axis is time, so 0 to 28 days. and then each of the panels is a species. And a Kaplan-Meier curve shows the probability that a seed germinates over time and allows us to compare germination timing among treatments while accounting for seeds that didn't germinate, so when we were unable to observe the seed between data collection times. And a curve that rises sharply indicates that many seeds germinated earlier, and a flatter curve indicates delayed or slower germination. Differences among treatments in the onset and steepness of these curves reflect differences in germination timing. These curves estimate the cumulative probability of germination for each seed over time while accounting for censored observations, so seeds that didn't germinate at all during the course of the experiment. Yes, and I could go through with you what the different lines represent, but it's basically different combinations of temperatures and dormancy breaking treatments. So the second part of the statistical analysis was the survival analysis part of my statistical analysis that I conducted was to get at germination rate. So how did the treatments and treatment combinations affect germination rate? And we did this by looking at Cox proportional hazard ratios. So the Cox mixed effects model estimates proportional hazard ratios, which compare the moment-to-moment germination rate in each treatment to a reference group's moment-to-moment germination rate. Basically, it allows us to compare the rate of germination of each species and treatment combination to a reference combination. So in this case, the reference combination was Allenrolfia occidentalis, germinated at a cool temperature. So that value is our 1. And if we had a hazard ratio that was greater than 1, that indicated a faster rate of germination over the course of the experiment. And if we had a hazard ratio that was less than 1, it indicated a slower germination rate over the course of the experiment. Faster is indicated by green. This will make more sense in the next slide. And slower is indicated by this warm color, this peach color. So here is a simplified version of the results for the hazard ratios that emerged. And we have a key up here in case you forget what the colors represent. But important to note is that each column is a species here. So when we're picking out patterns, it's most useful in the context of this talk to look at the species-specific effects because, spoiler, response to dormancy break treatment and temperature varied by species. Yeah, and that's what I will do. I'll go through some examples here. So these species, Allenrolfia occidentalis, Salicornia rubra, and Sporobolus oroides, needed cold stratification and germinated faster at higher temperatures. So at the top, you can see the moderate hot temperatures and then the warm and dry treatments crossed with the moderate and hot temperatures. And then the bottom half of this chart are the cold stratification treatments and the cold stratification treatments crossed with temperatures. These species, Disticula spicata, Glaucus meridema, and Salicornia utahensis, germinated to faster rate after cold stratification, but preferred cooler germination temperatures. So the Salicornia utahensis was actually germinated more slowly under a hot temperature after warm and dry treatment, and a moderate temperature after warm and dry dormancy break treatment, and just under a hot temperature on its own. These species, Puccinellia nataliana, which is a species I'll talk more about in the later part of my talk, Sesuvium varicosum, and Sueda calceoliformis were not dormant and mostly germinated more quickly under cool temperatures. So in particular, Puccinellia nataliana was slowed by both warmer germination temperatures and warmer germination germination temperatures paired with cold strat. In line with our predictions, responses to dormancy break and germination conditions varied by species. Most species required a period of cold stratification to germinate to a high proportion quickly. Some species germinated faster and or to higher proportions under warmer temperature regimes and in light. So from this, my recommendations are that we design seed mixes around the needs, the dormancy and germination needs of individual constituent species, and that we sow species that like cooler temperatures in the spring or fall, and sow species that require light to germinate quickly or to high proportions on the surface. Okay, and now I'm going to move on to talking about my field experiment. So how does the timing of seeding a playa and meadow-rich species mix influence native and invasive plant cover and composition? Seed-based restoration is essential for recovering native plant communities and attaining ecosystem function goals following invasive species management in areas where the native seed bank is depleted, like at Great Salt Lake. There's a critical demographic bottleneck that occurs at the seedling stage in plants, and seedlings are highly sensitive to environmental conditions. Land managers in the Intermountain West seek to revegetate wetlands. Typically, they initiate their restorations after peak snowmelt and as the water is receding. So in the early summer, late spring, or in June. But seeding in fall may be a way to overcome early plant life stage bottlenecks. The timing of seed sowing is a critical component of restoration that is within the control of restoration managers. So valuable to look at. I seeded experimental plots at 2 sites at Great Salt Lake at 3 different times throughout the 2024 season. Once in June, which is the standard sowing timing for managers. Once in August, which was our late summer seeding, and again, once in September. I seeded a 22 species playa and meadow majority seed mix for this experiment. So 16 of the 22 species were playa or meadow species. We also included 6 emergent species that are kind of known workhorse species for managers, and 1 upland species, the annual sunflower. And species spanned the continuum of hydrologic needs from facultative upland to obligate wetland. Seeds were dormancy broken according to their individual species needs for sowing in order to ensure high germination in the field. Our sites were located at Howard Slough Waterfowl Management Area and Farmington Bay Waterfowl Management Area. Both are highly managed places. For those of you that might know Farmington Bay, our Farmington Bay site was across from the Teal Lake unit, and the Howard Slough site was just outside of an impoundment outflow. Our 2 sites were hydrologically distinct. They were in different watersheds. The Farmington Bay site ended up being, as you'll see later in this presentation, dry for a majority of the summer. And our Howard Slough site received moisture throughout the course of the experiment over both growing seasons. This is my experimental design and my plot layout, block and plot layout at my sites at Farmington Bay and Howard Slough. This is just a simple map showing you the randomized complete block design. I randomized this layout and then manually adjusted for balance because it ended up being unbalanced after being completely randomized. And then zooming in here on just what a singular block looks like. Each of these colors represents a different seeding timing. So we left 1 plot unseeded as a control, and 1 was seeded in June, 1 was seeded in August, and 1 was seeded in September. Each plot was seeded at 5,814 pure live seed, which is 3 times the rate recommended as minimum sowing rate to managers. And then we recorded visual percent cover. I recorded visual percent cover by species at each plot type over 2024 and 2025 at 2 1 by 1 meter subplots in each plot. Yeah, so this is just more information on the water conditions at each of our sites. So these panels show groundwater depth. We installed groundwater wells at each block and recorded groundwater depth at each data collection day. In these plots, we have Howard Slough on the top, 2024 is on the left, 2025 is on the right. Those red dots indicate when I seeded in the field. And that horizontal dark line on both sets of plots indicates the ground surface. So as you can see at Howard Slough, water was above the surface of the ground for much of both the 2024 and 2025 growing seasons, while at Farmington Bay, there was an early season and late season plant establishment window in 2024, but for the majority of both growing seasons, water was well below the surface of the ground. That's reflected in what the sites looked like. There were like 12-foot cattails at my Howard Slough site, and by midsummer of 2025. And then the Farmington Bay site pretty much looked like this, and we could walk around it in sneakers for most of the 2024 and 2025 seasons. For my statistical analysis of data at this site, where we were not prevented from doing so by excessive zero inflation, I fit a generalized linear mix model with the beta distribution to assess the effect of seeding timing for levels on seeding timing and block on seeded native species percent canopy cover at the end of both growing seasons, total native species percent canopy cover, and total invasive species percent canopy cover at the end of each monitoring year. So moving on to my results. Generalized linear mix models of seeded species cover and total native cover at Farmington Bay. We couldn't fit those, so this is just plotted raw data that reflects what the site looked like. So there were pretty much no native species established at the site by the end of either growing season. And I'll just explain these plots for you. So the raw and modeled predicted mean proportional cover from 0 to 1 is on the y-axis, and then each panel is different years data. So blue is 2024, orange is 2025, and then we have our different treatments on the x-axis, and our seeded native species, total native species, and invasive species. We were able to model the effects of August seeding on invasive species cover for 2024 and 2025 at Farmington Bay, and August seeding reduced invasive species cover by 2025, surprisingly. But that might make more sense when we look at the raw data a little bit. Looking at the raw data, I just am zooming into 2025 here because it's the most interesting results that we had. It's like not that interesting to look at the 2024 data, but what's interesting about this is that if you look at these plots on the right-hand side of the screen, we have our different treatments, so the unseeded control, and then all of our different seeding timing treatments. And then we have pretty much 3 species that we're able to establish to above 2% cover at this site. Two of those were non-native species, Bassia hysopifolia, also called Kocha, Phragmites australis, and Puccinellia nataliana. And here, there's pictures of them. Bassia is also this big, dark. This is species cover these bar charts are species cover at the end of each growing season, and we're just looking at 2025. So that was the majority of the species cover by the end of 2025. But Puccinellia was a surprise, and maybe has something to do with what our invasive cover looked like at the end of 2025. The Howard Slough results, the 2025 native seeded cover model was fit using a generalized linear mix model adjusted for zero inflation. And June seeding reduced invasive species cover by 2025. June seeding had higher native species cover in both years, 2024 and 2025, but the effect was particularly pronounced by 2025. And June seeding had higher seeded species cover by the end of both 2024 and 2025. Our species composition at Howard Slough, we used the meta-MDS function to generate three-dimensional ordinations. However, the first two axes are displayed, but this visual separation of the ovals here is a real statistical effect. These different ovals represent the different treatments, and if they are far away from one another, that means that they're different from one another. And as you can see, the blue treatment is very different from the other treatments, and that's the June 2024 seeding at Howard Slough. So over both 2024 and 2025 species composition, in the June 2024 seeding was different than the other seedings. Our seeding timing results. So similar to our previous graphs, these axes are on the x-axis you have time, and on the y-axis you have 0 to 100% cover. And I'm just pointing out here that the June seeding, so this is the seeding that occurred in 2024, which is why it's labeled 24, but that can be a little bit confusing looking at the two different years of data. But it had high native seeded total cover in both years. Zooming in on what the species composition at the Howard Slough site was like over time and at the end of the growing season. These on the left are a little bit more interesting than the Farmington Bay stacked bar charts were. They show that there was much higher species diversity at the end of both years. And if you take a look at the 2025 panel, which is the second set of bar charts, you can see that second from the leftmost one in that panel is the June 2024 seeding. And clearly, native seeded species were making up a larger proportion by the end of that year than the native unseeded and the introduced and invasive species. So those species are represented by the blue color scheme. And then if you look on the right-hand side of the screen, you can see the different, over time, what the different species composition was in the different seeding timings. Largely, we had a lot of Hordeum jubatum at that site in 2025. But in our June 2024 seeding, we had quite a bit of Chenoplectus acutus, which is the heartstem bulrush. We had quite a bit of Chenoplectus americanus, which is the only three square. And then in our August and September and our unseeded control plots, a lot of Typha, which I mentioned before. So which species did well at our sites? This is exciting because a lot of these species have not been successfully established from a seeding effort before, the playa and meadow species at least. Some of the work species have been established before, so the Chenoplectus species and the Eliocorus are not too much of a surprise, and the Distichylus is one that is used frequently, and the Bulbochinus by managers. But the species that I think I'm most excited about is the Triglocan marittima. We also have Rumex maritimus, which is golden dock, and the Biden species, which is an annual forb. And then I also gave an honorable mention here to Puccinellia nataliana in the upper left corner, just because it was able to establish just some level of cover at our site, which was largely dry at the surface of the ground throughout the majority of the growing season. So overall, the big thing I learned is that field sowing has to happen in places that have adequate water for the establishment of seedlings. And it doesn't matter if you only have water at the beginning of the growing season, you need sites that have sustained moisture. And at sites with sustained moisture, seeding in June may establish native plants and suppress invasive species. I also think it might be worth exploring fall seeding of summer reproducing non-dormant species in areas with spring flooding. So if you have a drought-stressed site that's flooded during the winter because water availability is greater during the winter generally at the impoundments, it might be worth in the future expanding on research on that. And I also want to point out to managers on the Zoom or in the room that Nuttall's alkali grass, which is the Puccinellia nataliana, established and outperformed inland saltgrass, which is the Disticus baccata, that is widely seeded at Great Salt Lake at our Farmington Bay site when it was seeded later in the year. So my research impacts. I was hoping to inform native plant growers in their first step of growing plants, which is dormancy and germination requirements. So I identified dormancy, breaking, and germination conditions that maximize germination proportion and speed. An example of this is I identified several species that require cold stratification or light to achieve high or fast germination. We hoped to improve the understanding of how seeding timing affects plant community establishment and restoration, especially for these understudied playa and meadow species. I identified how sowing timing affects species establishment across sites with different water regimes. And at the site with sustained water, these playa and meadow species, as well as Eliochorus palustris, establish well under spring sowing and suppressed invasive species, while at the drier site, Puccinellia nataliana, established primarily under fall sowing. And I hope to facilitate the incorporation of greater species diversity into restorations. So I provided insight into which species to establish under different field conditions and demonstrated how sowing timing influences establishment. We found new meadow and playa species that established to relatively high cover levels when sown at a site with sustained water in June. So that includes Triglocan meridema, pictured here, Rumex meridemus, golden dock, and Biden species. I hoped to inform species selection for restorations occurring outside of emergent wetland environments. So field testing the playa and meadow species to assess their suitability for non-emergent wetland restorations is a start for that. And we found that several meadow and playa species established successfully. With that, I would like to thank my funders, the Utah Department of Agriculture and Food, the Utah Forestry, Fire, and State Lands, Utah Division of Wildlife Resources. Thank you to Keith Hambrecht, Rich Hansen, David England, and Talon Robinson. Thank you, Karin. Thank you to members of my committee, Ari and Scott. And thank you to all the members of the lab. And also thank you, Alice Carter, for your help with the statistics. Anyone have questions? Yes.
Were there, I guess, morphological characteristics of the seeds that helped you predict or think about if the cold stratification or what temperature would best be germinated at?
I wasn't looking at data of seeds, but we chose the seeds because they were common wetland, meadow, and playa species, and we were able to get of them. Yeah. And then just there wasn't like basic information known about them, and there's no like predictive framework for figuring out what what you need. So this was the first step in figuring that out. Yeah, that's an interesting question. Yes.
I only learned recently that plants need water. Demonstrated that
point.
at Farmington Bay. Please speculate about what might have happened if that were irrigated. And, I mean, because the water table dropped to a meter down low, below the ground surface. If you irrigated it for 1 year, for 2 years, would you then establish a sustainable crop of native vegetation?
I don't know, possibly. One thing that I did forget to mention is that the Puccinellia nataliana, the grass that I talked about a couple times, in July of this year, it had produced what looked like viable seed. I don't know if the seed is viable, so even with the water conditions as they are, there's potential for that to be able to reseed. But yeah, I imagine that applying water to a site immediately following seeding or at a point when the seedlings are vulnerable and beginning to establish would potentially allow them to establish. Though I was reading recently about how drawing down water at a site can stimulate germination from the seed bank, but you only want to do that for like 1 year, or it's so drought stressed that most of the seedlings die. So I think it's a fine line. I imagine that if the water were available to do that, that would be a good way to establish playa or meadow species.
Mature plants then could survive.
Possibly. I've seen species like, I mean, I don't know that, but Triglockin meridema I've seen growing in really sites that are dry. I think it was maybe like a spring mound, so the groundwater might have been closer to the surface at that part, but at that part of where I was. But maybe some of them, the meadow species, I mean, they are still wetland species, so they need some amount of water. They're not going to grow like, I don't know, on pavement. But yeah, I have seen irrigated restorations that were established with plants, so like plugs that are doing well under like standard lawn irrigation conditions that are all wetland plants. So yeah. Yes?
Nice talk. Could you go to your slides where you had, it's not the survival analysis, but the germination time?
Let me know when to stop. You're talking about the Kaplan-Meier curves.
Yes.
Yes?
Yeah. So on a number of these, you see right at the very end, there's kind of almost like compensatory germination going on. Is that where they seem to hit some plateau and all of a sudden right before the end?
I think that's an artifact of the model more than it is the reflecting the actual germination dynamics. Yeah, but that's a good question. I believe that where it flattens out kind of is more reflective of what is going on with the.
Because I don't know, it's like that's what fish would often see. Slow growing fish will catch up at the end of the year. Or somebody.
Yeah, well, looking at the raw graft germination data, it doesn't look like it does that as strongly in most of these species. But this approach is a way to model all of the different species and different treatment responses without like choosing from a bunch of different models and then cherry picking like which one works best. So that's why I chose this way.
I'm just going to very next.
Yeah. Yes.
So you were saying for your methods for the field seeding,
Mhm.
dormancy was broken into individual species needs
Yes.
for using them in the field, and I thought like, what a luxury thing.
Right.
And so would you recommend that to take the time for, managers like myself to do that because it's worth it? Or is there maybe some intermediate strategy that could work as like a blanket thing that you can kind of rely on for the full stratification?
I would say if it's possible to dormancy break seeds that have similar needs together, do that. But a couple of the species germinated at least more slowly following dormancy break. So if you have data on that, like you would want to avoid dormancy breaking the Puccinellia or any species that you know are non-dormant. Yeah, I would recommend dormancy breaking them individually, but there's totally wiggle room in some of them. So some of them respond well in terms of increasing their germination proportion or increasing their speed of germination following like 4 or 8 weeks. If you only have 4 weeks, maybe put all of the ones that can do 4 or 8 weeks together. I don't know. How are you guys doing it in big tubs in different packets or?
So I personally haven't done it since I've been with FFSL. The colleagues that I work with do. But yeah, I think it's kind of like a broad stroke approach. And then there's obviously like the 50-50 half and half. But yeah, that's something to think about, like not just assume that even though physiological dormancy is common, don't assume that they all need it because they can actually use it.
Yeah, I think if you have data on individual species and you're able to separate them, then that's the approach that I would take. I know that there's, there are limitations that field managers have that I might not be aware of, but that's what I would do if I had the ideal.
We have the extension document, obviously, that myself and others can look to.
Right.
And
yes, and some of this data went into our new seed dormancy break document as well. So some of that data is out there, and I would recommend looking into the species that you're working with and following the recommendations that our lab has come up with through studies like this and others. Yeah, thank you. Anybody else? Yes, Tim?
In your field experiment,
Yeah.
for the August and September sowing, you may have said it, I might have missed it.
Okay.
Did you clear those plots the same time before seeding as all the others?
No. So we were limited by what we were operating at a fairly high our spatial scale was pretty large, so we couldn't just hand pull stuff. But it was so small that we couldn't have managers come through. So we decided to just go with how they prep the site in the spring and then test the effect of seeding timing on that. And that's an interesting question. I think our cover was our total vegetative cover was pretty low at the Farmington Bay site throughout the course of the experiment, including when seeding in 2024. So I wonder if that allowed seedlings of at least that one species to be able to establish from the later seedings. Yeah. And also going back to your question on irrigation, at the very beginning of the experiment in 2024, following our first June seeding at the Farmington Bay site that was dry for a lot of the season, we were seeing seedlings of plants that we had seeded. So water probably would have helped at that point. They got crispy pretty fast. Yeah. Okay. Anyone else? Any questions? Okay. Thank you.